So welcome everyone to our weekly AATRN seminar. The speaker today is Kelly Max of EPFL, and he will talk about cohomology classes in the RNA transcriptome.
All right, so thank you to the organizers for having me. Today I'm going to talk about some mixture of algebraic topology and biology, and it's definitely one of the more applied projects I work on. Importantly, this is joint work with some people on the call, so Marcus and Tam in particular. I want to give a special thanks to a couple of biologists that we've started talking to more and more, Cyril and Wouter at EPFL, and my supervisors, Catherine and Catherine, right? So yeah, maybe an important point before I start is that I really want to keep this talk, because it's applied very low on notation, as low as we can get, and keep it more at a conceptual level. And what I really want to argue is why cohomology is the right thing to do, right? More so than cohomology works, but I want to give a more philosophical argument of what cohomology really means here. So at a high level, the goal of this project was to identify and exploit circular structures in single cell data. But the step in that is really presenting a kind of cohomological interpretation of biological processes, and in particular, something called a closed biological process that I'll define in a bit. And the idea is that if we can understand, in some sense, the kind of biology we can look for, that will help us pick data sets and understand what the data is saying. And in particular, we're going to develop tools with this intuition that will locate circular geometric features and kind of somehow understand which genes and various biological things are contributing to the production of those circular features, right? So why can we talk about kind of genomics and topology and geometry in the same sentence? And in some sense, there's this overarching concept of the genomic phase space, right? So this is maybe a conceptual idea, a very old conceptual idea in biology, is that somehow, if we have a cell and genes, the genes express themselves by making copies of RNA, right? So you can think of the RNA as a measure of the gene's activity in a cell, right? However many RNA molecules corresponding to that gene there are at a given time. And that somehow there's an extremely high dimensional space consisting of all possible gene expression configurations, right? So this is sitting in something, roughly 20,000 dimensions of the genes, and somehow the cells are sitting in this space in some really complicated way. And the underlying principle is somehow that the geometry and topology of this space corresponds to biological hypotheses and ideas. So let's give a really simple example. Imagine I have two different types of cells. Well, the idea is that their gene expression will be really different and isolated in different islands. So basic topology idea is that these cells will somehow cluster in the phase space. But, there's really old ideas in biology that correspond to more kind of continuous processes. So this is a really famous picture in biology called the Waddington landscape. The idea is that you really want to visualize the differentiation as a cell. You think of the cell as a marble on some sort of geometric surface. And as time progresses, the cell's gene expression changes according to the kind of geometry of this surface, right? So in some sense, we can see developmental pathways. We can kind of think of them conceptually as kind of geometric properties of the phase space. Now, there's something that kind of directly contravenes this idea of, kind of linear differentiation. And that kind of geometric structure, I think, is really captured by this Escher painting where, imagine I start somewhere, I go around, and instead of rolling to a different kind of cell state, I roll all the way back to where I started. And I going to kind of call these, when this happens in biology, a kind of closed process. It's like a closed loop. I start somewhere, I go around, and I come back to where I started. And as topologists, the kind of deep intuition is that this is something kind of topological. This is some sort of loop structure, and this should be measured by some sort of topological invariant, namely something like cohomology or the fundamental group. So the kind of hypothesis of our work, and I think it's, maybe not stated this way, but it's kind of out there in biology, is that these kind of processes that start, go around, and come back are abundant in biology, right? So there's heaps of them. And the kind of canonical example of this is the cell cycle. So cells, when they divide, they start at some state, they go around through different phases, they divide, and the divided cells look roughly the same as when you started, right? So exactly like this, the cell, goes into this cell state, goes around this process, divides, and it looks basically the same as when you started. And there's other things that are periodic that kind of oscillate in waves over time indefinitely, like the circadian rhythm. Our gene expression is kind of known to oscillate with the kind of hour of the day, right? At noon, it looks like kind of roughly the same, changes, comes back. So these were the kind of canonical examples, if you look in the literature, where people are thinking of kind of closed biological processes. But I mean, what we found, and we're going to go into in the talk, is maybe a third type, is when a kind of biological system has some sort of base point homeostasis that it's kind of wants to be in at all times. Often some external stimulus will kind of make that biological system deviate from homeostasis. And then somehow there'll be some sort of biological gravity that brings it back. And if the kind of stimulus genes are different enough to the response genes, then this is also going to cause some sort of loop structure or closed biological process in the phase space. So geometrically, there's been this idea floating around for a really long time, right? Biologists often draw these pictures and think about things geometrically, but in the 21st century, this became more literal and more computational, right? So people started really thinking, okay, how can we actually estimate and model the kind of geometry of this phase space in a computer and with kind of actual sequencing techniques? And in some sense, single cell data is some sort of finite sample of this giant kind of theoretical genomic phase space. So what they do in biology is really amazing, right? They get a tissue, they put it in a big blender, get all the cells apart, and they have this amazing way of, you get all the RNA molecules in the total tissue, and you can kind of reconstitute them or kind of estimate which cell they came from. And, this is kind of complicated technology, but what I get as a mathematician is really just a matrix that has the kind of cells and the genes and however many RNA copies of the cell of each gene are in each cell. And roughly, we can really just think of this as a point cloud, right? That is some sort of estimation of our big phase space. Now, one of the key points is that this is kind of, once you do this to the cell, you destroy it. So you have a finite approximation. But not only that, you have a snapshot at one given time, right? So what you kind of need to do is estimate the genomics, the kind of dynamics of gene expression from the structure and geometry of this phase space. So let's see, kind of conceptual examples. They have analogs in single cell sequencing. Really, when you look at a single cell experiment, often if you do like a UMAP plot or something, you really see the dominant thing is clustering by different cell types. But interestingly, we can also see these more continuous or smooth processes, right? You start with some sort of like pluripotent or stem cells, cells that haven't decided what kind of cell they want to be yet. And you can see kind of through a sampling of the phase space that cells have this kind of tree-like structure of cells evolving over time. And even though we just have one snapshot, because we're capturing cells at different points in this kind of evolutionary process, we can really see the kind of continuous part of this phase space that represents some sort of developmental trajectory. And further, we see these kind of closed biological processes that we're interested in that somehow correspond to some sort of cohomology, right? Though that's at least the kind of geometric thing that we're seeing. So here we just have a cell cycle. You can kind of see G1 goes into the next phase of the cell cycle and finally ends up in the last phase. The last phase looks very similar to the first phase, right? So we really see this stuff in data. It's very geometric. So for the rest of the talk, we really want to focus on this third process, these kind of closed things. And we're going to explicitly use geometry and topology to really generate biological hypotheses about these that correspond to these processes. And we're going to introduce two geometric tools. One is circular coordinates, which people in TDA, I imagine, are pretty familiar with. And the second is something called cyclicity analysis. So kind of this underlying question that we've hinted at already is, what is a good mathematical signature of these closed biological processes? And for me, really, we really just want to focus on giving some sort of cohomological formulation of these guys. So maybe let me start with a slightly different definition of cohomology than the algebraic one that is maybe more obvious why this is a good model of a closed biological process, right? So when we have a kind of single cell experiment or a closed biological process, what we want to do is kind of assign a kind of circle value to tell you what point in the closed process you're on, right? So in some sense, a closed biological process should be represented by a kind of continuous circle valued indexing, right? Or a continuous map from the phase space into the circle. But obviously there's, uncountably many of these. So you want a kind of natural notion of equivalent biological processes. And that should be something, at least topologically, is base point preserving homotopy classes of these circle valued maps. So immediately that destroys kind of trivial examples where you can kind of contract the circle valued indexing down to the base point as we are here. So if we start with our we're trying to model these equivalent closed biological processes, but we want to do it with homotopy classes of circle valued maps. Well, a kind of standard theorem in algebraic topology is that these homotopy classes of base point preserving circle valued maps are kind of classified by the integral cohomology classes in dimension 1. And if we really want to understand these kind of complicated homotopical objects, we can understand them at the algebraic level. So, importantly, if we're just starting with a point cloud, that doesn't have any notion of topology, and we don't have a notion of what the continuous indexing is. So the standard TDA answer is to replace our point cloud with a RIPS filtration or a check filtration here, which is in some sense looking at kind of different topologies to kind of have a different kind of notions of closeness at different resolutions. And a summary of all possible kind of cohomology on this is the persistence diagram, cohomology classes that are born at some time and die at a later time. So I guess the triangle of ideas here is kind of important, not so much in a mathematical way, but linking it to the biology is that what does kind of persistent cohomology mean in this setting? Well, in some sense, that's looking at kind of equivalent closed biological processes at every possible resolution. So in some sense, when we compute our persistent cohomology, we have a summary of all of the possible kind of closed biological processes, and roughly the persistence quantifies their geometric or global importance. Now, an old idea that's kind of fundamental in the circular coordinates algorithm is that to each kind of persistent cohomology class at a given resolution, we have a kind of canonical co-cycle representative that represents not just the topology, but the geometric information. Right? So when we start with a point cloud, we move from the point cloud to our kind of algebraic summary. And the circular coordinates algorithm is, in some sense, we start with our kind of persistent class and we're representing it as a kind of circle valued map or a kind of one co-cycle on the space. And I think a good way to think about this is maybe thinking about it in a more kind of Riemannian geometry way. If I start with my kind of smooth space or Riemannian manifold and take a circular coordinate, somehow I can look at the gradient of that circular coordinate, right? Imagine I have a gradient vector field that is locally the change in angle. The harmonic representative is somehow the smoothest kind of circular map in terms of what has the smoothest possible gradient, right? So in some sense, the kind of canonical part of this is that we're minimizing some sort of energy functional, right? Which is the kind of smoothness of the gradient. And what the circular coordinates algorithm is really doing is doing some sort of a simplicial approximation to this Riemannian story, right? So the kind of dictionary is the de Rham complex in the Riemannian world corresponds to the simplicial cochain complex. My kind of one form, or this kind of dynamics representing the dynamics of the circle valued map, well, that's represented as a one cochain. And somehow the Riemannian metric, which is some sort of geometric property of the space, is represented as an inner product on the cochain complex. And the kind of key technologies here that already exist in TDA are, in some sense, this conceptual idea going all the way back to the persistent cohomology and circular coordinates algorithm. But importantly, in single cell stuff, we have highly non-uniform density. So we use a kind of modified version of this, going to in this kind of really interesting paper on kind of density robust circular coordinates, where they rescale the inner product to account for density. And thirdly, in our single cell experiments, we have kind of giant point clouds and we need some sort of sparse subsampling. So we use all of these kind of existing TDA technologies and methodologies to deal with the kind of inherent problems in single cell stuff. So in some sense, the utility of this definition is kind of, it's only useful if we can actually find these guys. So, the first, or maybe the first kind of 6 months working on this project, the hard part was going, okay, can we actually see these guys in this extremely noisy and screwed up data? And maybe the fundamental problem or way to think about this is that actually there's heaps or shitloads of different biological processes happening at the same time, and they're all twisted together in the phase space, right? So you can kind of imagine if I just have two simple different things happening, I have this Waddington landscape, which is that cells are kind of evolving over time, but maybe orthogonally to that, they're kind of rotating around some sort of cell cycle or closed biological process, and they're kind of twisting together in this kind of bundle or like product space. And what's really interesting is that you actually see this a lot, right? And you see that when people get like the whole gene expression space and do a low dimensional projection, you can see that in some spaces, maybe the cell cycle part or the circular part is dominating, and in other spaces, it's more a differentiation thing. And when we try to kind of compress all that together in a low dimensional rotation, well, we're kind of squashing this product in different ways, and we need to somehow it's trying to do too many things at once. So maybe the right way to think about this is that if I have this kind of theoretical phase space embedded in my kind of ambient gene expression space, what I want to do is find a low dimensional projection where the data really looks like a copy of S1, right? And in some sense, that projection should correspond to the kind of circular coordinate, right? And if these things play nice together, what this is really trying to do is split off a kind of S1 copy of my product space, right? If I can find a low dimensional subspace where the data is S1, well, maybe this is finding a kind of component in the product of this giant global phase space that corresponds to the product of many different biological operations. So our mission and goal for constructing this is to find subspaces that correspond to closed processes, right? So here we do something highly non-canonical, but more biological, which is what's a kind of, obviously there's millions and millions and billions of different subspaces to check. Maybe a good space to start is to start with subspaces that consist of genes that correspond to some sort of biological process, right? So biologists love these gene sets, right? Like they've spent years and years and years curating gene sets that mean that where the genes correspond to some sort of biological process. So maybe what you'd want to do is just search through these kind of hallmark gene sets and see if there's any circular information in there. So because we have this kind of connection, because we're really thinking of a kind of closed biological process as something geometric, we can essentially just take each kind of gene set, do a low dimensional PCA thing, and see if we have non-trivial persistent cohomology. And this would be a hint that in this gene set, we're seeing some sort of closed biological process. So for us, we define a kind of basic ring score, which is a kind of statistical summary of the persistence diagram, essentially answering the question, is there a kind of dominant circular structure, right? Does my kind of low dimensional projection look like a circle? And we can do things like permutation tests to get a p-value for this ring score. But importantly, we can automatically sift through gene sets looking for circular structure. So let's have one example to see, does this work or do we just get kind of crappy numbers that mean nothing? So when I first gave this talk, I talked about the cell cycle, and afterwards something amazing happened. A biologist, Wouter Carhaus at EPFL, came up to me and he said, I've got this old data set and I think this is the kind of perfect test, right? Like what we do, we have something that's exactly a closed biological process. If you can't see it in this data set, then I feel like your method's not going to work. So this was let me give you a kind of idea of what's happening in this biologically. What they do is a little bit cruel. You start with 11 different mice, you castrate them, you nip out their prostate, right? And you just kind of let it sit for a month, right? You let everything deteriorate, whatever happens, happens. And what you do is you add in these hormones. You add in testosterone, which triggers androgen, and slowly the prostate regenerates over a month, right? So you have this process of degradation. And then somehow after this, the prostate magically regrows back and it's in theory meant to look like what it did when you started. And in particular, we have 11 different single cell experiments joined together. So we have a ground truth notion of time. And we're really looking to understand specific gene sets, right? So in some sense, what we're trying to see is these specific luminal cells, how are they regenerating in the process of time? And this is one of these processes I talked about earlier. You have some homeostasis where the mouse is just normal state. You do something horrendous to it, right? And all these stimuli genes respond to adapt. And then you inject it with something and it triggers this process of a return to homeostasis. So something amazing happened when we loaded up the circular enrichment. the thing that we were expecting to happen, the androgen response, that was the most circularly enriched score, right? So somehow the geometry of looking at this subset of just the androgen part, we can see some sort of circular geometry. So the question is this circle the same as this circle from the paper? And amazingly, it is, right? When you plot the kind of circular angle as you go along, you see that the kind of histogram is that, at the start, we have this kind of intact castration point. And as we go along, we're seeing the kind of underlying ground truth develop kind of in proportion to the circular coordinate. So this was kind of promising for circular, this kind of notion of, okay, being able to find geometric features that really mean something. But in some sense, this is still qualitative, right? What we really want is we want to unpack this. We want to take this geometric feature and we want to analyze the shit out of it and understand and unpack specific genes and biological signals that are contributing to it. And this is where we have to go deeper into the actual geometry of this cycle rather than just the topology. And maybe I call this, if the previous section was kind of searching just for topological signals, now what we're trying to do is recover the gene cascade or the order in which genes are happening in that topological signal, right? So in stage one, we're finding these kind of geometric structures, but the idea is that there's going to be some sequence of genes that are kind of pinging or peaking along this topological structure that are the reason that this topological structure exists in the phase space, right? So you can kind of imagine when I get to the blue part, there's some blue peaking gene. When I get to the green part, a green one, yellow, yellow, and blah, blah, blah. So the kind of question is, how can we kind of start with the phase space and go back into the ambient space to estimate the gene cascade? So there's a tool that was developed by Baryshnikov and al. In 2016 that if you squint looks exactly like the thing we want, right? So the difference is that what they start with is a closed path, right? And what they're trying to do is analyze somehow how these closed paths interact with the dimensions of the path in the ambient space, which is roughly what we're trying to do. So imagine here, I've got a path, and let's think about this as a trajectory in a two-dimensional gene space where I have a blue gene and a yellow gene. They start off, something happens, the blue gene is triggered, then the yellow gene is triggered, and they go around, they come back to where they started. So if you kind of plot the kind of the blue gene against time and the yellow gene against time, what you see is really interesting. You see that one gene is kind of firing off, and then the second gene is firing off almost as a kind of shifted copy, right? And in some sense, this is our model for one gene kind of controlling the expression of the next gene, right? And the kind of vocabulary for this in the Baryshnikov paper is that the blue gene is somehow leading the yellow gene. But the idea is that, we don't actually need to see the blue gene and the yellow gene. What we need is some sort of, this is happening in some sort of geometric structure in the phase space. So maybe the idea is that if we look in the phase space, that somehow this kind of, this path looks like a flow line of angular velocity in some sense. And indeed, if you integrate this path against angular velocity, if I kind of look at the kind of integrated correlation of the gradient of this path against angular velocity, that precisely corresponds to the kind of signed area of the curve in the plane, right? And what this is doing is kind of giving us a statistic that tells, is this kind of blue gene leading the yellow gene? So if we want a kind of concept of lead lag in our phase space, we need a kind of way to we need a stand-in model. What should our kind of model for the path be in the phase space, right? Now we have no intrinsic notion of time in our single cell experiment, so we need to do something else. So the idea is somehow that we use our circular coordinate as a kind of synthetic model for time, right? So let me maybe explain what I mean by that. Somehow the circle is a kind of canonical closed path. We don't quite have a circle, but we have a map into the circle. Maybe instead of the gradient of the path, we just kind of pull back the kind of gradient of the map, right? So in some sense, we've estimated this kind of circular structure in the space. We take the kind of harmonic vector field, this thing that is kind of maximally smoothly changing along the angle, and we use that as a kind of model for a likely dynamics, a synthetic likely dynamics on our phase space. And what we can do, maybe ignoring a little bit the annotation, is that take this kind of canonical form that models rotation And we can just compare it with the kind of rotational vector field that we've estimated from our kind of cohomological structure of the phase space. So maybe in a kind of more philosophical way, what we're doing is we're looking at our kind of projection into different pairs of genes, and we're seeing, are we seeing this kind of rotational structure of one gene leading the other in each pairwise plane? And what you want to do is assemble that into a matrix where you can kind of see all this pairwise information of gene I leading gene J and blah, blah, blah. Now, the lesson is that whenever you assemble something into a matrix, the spectral information is going to do something magical, right? And this is maybe an idea that goes back to the Baryshnikov paper, is that somehow if I kind of take all this pairwise information in the lead lag matrix, do some sort of spectral thing, what that is going to do is kind of assemble all my kind of information of which gene is in front of the other and spit out a kind of global ordering. So this is the spectral approach taken by Baryshnikov and al. This is not original. The original part is kind of getting a lead lag matrix in this setting. So in some sense, you have this skew symmetric matrix. When you diagonalize it, you get a complex plane, and you can show that the kind of principal complex plane there is kind of maximizing the rotational energy of our circular coordinate or our kind of simulated dynamics. And more than that, if you look at the kind of complex eigenvector and look at the components of it, that's giving us a kind of estimation of which genes are occurring before each other, right? And the global ordering on them. And this is exactly what we want, right? We're starting with our kind of topological feature, this kind of circular coordinate that gives us some sort of model of the dynamics. And we're going all the way from that into a kind of estimation of which genes are contributing to that in which order. And the way I think about this is some sort of cyclic or kind of rotational PCA. In PCA, we look at a covariance matrix. The eigenvalues give us the covariance, and eigenvectors are kind of maximally covariant dimensions. In lead lag, we start with this lead lag matrix. We get lead lag coefficients, and somehow those guys correspond to the kind of rotational energy in each plane. And now if we go to our prostate example, right, and we do our lead lag projection, we see something really nice. And we see that when I do this kind of linear projection, this projection kind of is coherent with our circular coordinate, right? Like somehow we're finding a plane that kind of maximally looks like the rotational part of the circular coordinate. And you can see that as I go along the circular coordinate, I'm kind of seeing the ground truth time develop in a way that makes sense. So even more than that, we can estimate our kind of gene cascades. And what you see is when you plot these gene cascades against the coordinate, we're kind of seeing the realization of this conceptual picture, right? We're seeing this kind of starting at a point, deviating from homeostasis and coming back. And we're seeing a kind of continuous wave of gene cascades kind of spiking like neurons in order. And we're seeing the kind of phases develop along the circular coordinate. Plotting in another way, if you just really plot those genes in order, you can see them kind of pinging along the circular coordinate as we move along, or along the kind of different things. So we're really seeing those gene cascades without any intrinsic notion of time, but in some sense having a kind of cohomology dictate a kind of likely notion of time relative to this. So not only do we see that, I guess maybe the biological hypothesis was that, you know, these guys are abundant, and let me show you two more examples, starting with the cell cycle. Where we can really see this stuff happening, right? So we wanted to start with an example where people know that there's the cell cycle, right? So we have this data set from some colleagues at EPFL with cycling human dermal fibroblasts. And we're expecting to see somehow this picture. And we know that this data set has the cell cycle because there's a paper by some colleagues at EPFL where they've kind of done the analysis. You can see the cell cycle geometrically. We know it's there. So if you do this kind of detection in cell cycle space, find the circular coordinate, look at the eigenprojection onto this plane, you see that the different cell cycles, right, as kind of indexed independently, or the phases, they're kind of moving along this plane in a way that you expect, right? We see a kind of the S phase moving into the G2M phase, and these guys are consistent with the kind of estimated circular coordinate. What's more, if you look at the kind of this kind of estimated gene cascade, you can see all the genes in the S phase occurring before all the genes in the G2M phase. Somehow in matrix form, you can see that all the genes in the S phase are leading the genes in the G2M. But actually, within each phase, there's different genes leading each other. So there's more of a continuous resolution of genes happening. And when you plot them, you get these really nice kind of continuous waves of peaking gene cascades. In some sense, we can see that genes are occurring in some sort of global order relative to this circular coordinate. So it's extremely geometric, and the gene cascades you get from the geometry really agree with the biological interpretation. So the final example is one that is somehow something that is maybe going a bit out there with something that I think that maybe biologists don't think of as a closed process normally. And this is something that maybe we saw empirically that came from the geometry and then later we got a biological interpretation for it. So the data set is an extremely standard data set, right? We used it because it was the easiest to access and because this is something that people have analyzed a lot. It's in a tutorial for standard processing pipelines. And we reasoned if we could see something interesting in this data set, then somehow our tool would be actually adding value to something people have already studied a lot. And the idea is that we're looking at these hematopoietic progenitors, which are in some sense just blood cells. And when we load it up, so it's a bit of context, people use this for kind of identifying the cell cycle. And when we loaded up the circular gene set enrichment, we saw something really interesting in that we saw things like interferon alpha response, apoptosis, things not normally associated with the cell cycle were pinging higher than traditional kind of cell cycle marker genes. So this was, I guess, concerning as a mathematician because you're expecting to see something biological. You have no biological intuition. So the two questions were, okay, we're seeing the cell cycle stuff. Is that transferable to other examples? And the second one is these kind of immune responses, are they signal or are they some sort of geometric noise that are showing that our method isn't working actually? So starting with the cell cycle part, right? The picture is very similar to the kind of previous cell cycle example. You know, I plot it down into this principal plane. I can see the coordinates developing. And the coordinates develop roughly, you know, as the cell cycle develops, right? As I move along here, I see one phase of the cell cycle continuously move into another and come back. And I see my S genes and my G2M genes. And actually, you know, I've got one data set where I'm looking at the cell cycle. I've got another one. If you look at the kind of two estimated gene orderings, they're relatively similar, right? Like I look at the kind of estimated gene orderings here and the estimated gene orderings here, and I'm seeing a really nice correlation, right? So somehow there's some degree of transference between these two data sets. But the real question was, why are we seeing these gene sets, right? Like, what does it mean to see this immune response? Is our kind of method broken? And I guess here we had to really talk to the biologists to see, you know, can we validate this by saying there's a biological interpretation by what we're seeing? So let's start with interferon alpha response. Interferon alpha is some sort of thing that, you know, like it's a molecule that when cells are stressed, they kind of emit this little particle that tells blood cells to come and do stuff and fix the problem. And when we look at the kind of projection here, we see this like highly non-uniform density. And what we're doing is we're getting some sort of geometric prediction for the order in which cells are happening in this process, right? The order in which genes are occurring in this process. So this is a purely formal construction based on the kind of underlying topology of this, the persistent cohomology, and the purely formality kind of these integration and just popping out these genes, right? This is just totally mathematical, no biological knowledge whatsoever. So our colleague Tam, who's fortunately a mathematician and a biologist, looked at these gene sets and was like, you know, this actually makes sense biologically, right? Like maybe I look at the first genes, this cell and LY6E, right? And what these correspond to is that when interferon is kind of emitted, these kind of neighboring blood cells in the blood vessel, this is kind of what's calling them over and inducing proteins on the cell surface that allow the kind of cells to kind of smoothly migrate to the site of inflammation. So this was maybe the first thing you would expect to see when the interferon is emitted. And as we go along the process, we get these kind of different estimated genes. And you see things that kind of correspond to later processes biologically. So here, the later process is what's happening inside the cell is it's going through a process called ubiquitination. In some sense, this is when proteins internal to the cell molecule, to the cell structure are being tagged, and it's saying, okay, I'm going to destroy these proteins in preparation for the cell to be destroyed. So maybe the main point of this is that the kind of formal geometric predictions that we're getting actually are coherent with some sort of biological intuition, which was somewhat surprising to me. So the late phase of this is super interesting, I think, and maybe this is like the most promising direction of future research is that, you know, what we saw in the immune response was two different processes, right? We saw this interferon alpha response corresponding to this geometry. But we also saw some sort of apoptotic gene set enrichment. And what happens is that when I enter this kind of late stage of the interferon alpha response, the cells need to make a decision, right? They're kind of been cooled over, the immune response is happening. In this immune response, are my stress cells going to die? Are they going to enter apoptosis? Or are they going to divide and differentiate and kind of escape the kind of death process? And if you look at the interferon alpha response coordinate, right, this kind of time coordinate versus the apoptosis coordinate, you see this really clear bifurcation, right, at this point, where somehow the gene that's a signature of apoptosis is kind of broken away that's from a scene that's a signature of mitosis, and you can see a kind of decision boundary of the cell going, okay, am I going to die or am I going to live? So somehow the pure geometry of this, right? Like that you really get these like very interesting geometric features purely from a formal perspective. And then the game is being like, can I validate this with biological knowledge? So you have to go back to the biologists and be like, what does this geometric feature mean? And so far, having conversations with biologists has been really exciting because they go, oh, of course, like, you know, this gene, this is like, they see that and they immediately have intuition for what that means biologically. So maybe as a summary, right, like this notion of closed biological process is some sort of geometric one, right? There's this very old idea that, you know, biology, really, you can think about a lot of it in a geometric way. And what we're doing is taking a lot of tools that already existed in TDA, right? Once you formulate the problem as a cohomological definition, then everything you have for calculating cohomology and estimating cohomology, you can actually use and interpret, right? So there's this first step of really having a biological theory of what the cohomology means. But then after that, we have this mature field of TDA now where we can really calculate these things and get really good estimates and use these tools. The key points is that this single cell data is really interesting for topologists and geometers because it's really topological and geometric. The reason that TDA is useful is because the cohomology has some sort of biological meaning. And third, we don't have to use any machine learning here. We're really just using TDA and geometry to kind of back out the results. So yeah, I just want to thank my collaborators, the biologists I've been talking to, the lab, and obviously my sponsors here. So yeah, thanks guys.
Thank you. So maybe I would ask everyone to unmute themselves so that we can give a round of applause for Kelly's wonderful talk. And now it's time for questions. So feel free to raise your hand or just unmute yourself and ask.
I'll start with the question, Kelly. Could you remind me what the definition of this lead lag matrix was again, roughly?
Yeah, totally. So let's go back. So for a path, the idea is that you look at, you've got a path in Rn, right? You look at it, you project the path down into like ij for two different coordinates. And you essentially, it's a closed path, so you calculate its signed area, right? So the lead lag coefficient is the signed area of this closed path in a particular ij subspace. And the idea is that if you have like sine and cos, for example, in that dimension, that will generate a kind of circle, right? And it will have a kind of positive signed area. Now, the lead lag matrix is just it's kind of putting together all this pairwise information for each kind of ij coordinate.
And I and j are just different coordinates, like
yeah, literally
you choose a basis, or
you choose a basis, but because integration is linear, some sense, it respects change of basis. So if I do a unitary transformation of that matrix, that represents the kind of lead lag in the new basis, right? So in some sense, the diagonalization is finding a good planes where you kind of maximize the signed area, right?
Cool. All
Somehow,
right,
because
that's really nice.
yeah, the linearity of integration. I mean, this is the Baryshnikov stuff, but the linearity of integration means that you don't. It's somehow invariant, like it respects change of basis. Now, this works as well with integration over the phase space, which is really nice, right? Like if I have this differential form and I integrate it, somehow because the inner product is linear and the integration is linear, this kind of change of basis theorem holds over these phase spaces, right? So you can do the same trick.
Thanks.
And then we have a question from Bastian. Bastian, please go ahead.
Yeah, thank you for a wonderful talk, Kelly. I have a quick follow-up to that one. Is this by any chance related to the path signature?
Totally.
It seems awfully similar. Yeah.
Yeah, so I mean, in this paper, this is like one of the path signatures are these lead lag coefficients, right? They're in level 1. And that's in some sense what, I mean, when I thought about this, Derek, who's on the call here, who's an expert in path signatures, had been talking about path signatures a lot. In some sense, it's coming from that, right? The difference here is that we don't have a path anymore, right? So we have to use something else. And maybe that's the main innovation of what we do, what we add to it, is that instead of using a path, we can use some sort of harmonic representative as a cohomology class and get a kind of path signature, a synthetic path signature of that.
Okay, very, very nice. Thanks so much. That's great.
Yeah. Thanks, Bastian.
And we have more questions for Kelly. So there is one question in the chat. Have you compared the gradient of your circular coordinates with RNA velocity?
That's a really good question. So the paper I mentioned that it looks at the cell cycle here, this paper by Lederer and al. That's on bioRxiv at the moment, just came out in January. In some sense, what they were trying to do is a very similar thing. They were trying to find some sort of circle map to calculate the RNA velocity. And I guess the next thing we want to do is really look at comparing with their results with the kind of cohomological model to see if we're getting similar things. So that's a really good question, right? Somehow, like, is this actually recovering RNA velocity? And that's definitely one of the next things that we're going to try to, is to look at that.
And then we have so thank you. Alamin, do you just want to go ahead and unmute yourself and ask?
Thank you very much for the nice talk. Actually interesting on the application of topology on cancer. So sometimes we have like the cancer itself is heterogeneous. So when we give a treatment, then there's a response. But after a while, other cancer lineage will occur. So I'm interested on how we can see the topological feature of this phenomenon.
Totally.
Thank you very much. Yeah.
So actually the prostate regeneration example, the idea, to go back to cancer, the idea is that what Wouter was really trying to study was that this process after you give it the testosterone, right? The idea that the prostate regenerates, that's actually something that cancer does, right? Somehow cancer learns how to hijack the kind of prostate generation process as a way of kind of learning how to use androgen to replicate. So really, actually, what he was trying to study is like a normal function in the prostate with an eye to understanding how cancer hijacks that process. So the idea would be that if you have these heterogeneous cancer cells, maybe they're heterogeneous in the global phase space, but in some subspace, right? In this we can split off just the part that corresponds to the androgen response, and we can see, is the androgen response part look similar to the thing that we're seeing in the normal functioning? So even though cancer cells might be extremely heterogeneous, the idea is that if you can decompose the phase space into different processes, maybe you'll be able to find some sub-process that you can understand within the cancer, right? That's in some sense the motivation.
It's actually interesting.
Yeah.
Thank you.
It's heterogeneous, but there's so many different things going on. Maybe there's a part of it that's actually very homogeneous, right? Maybe you can split off a sub-process, and that's something you can actually tangibly understand. And in some sense, this is what we're trying to do.
Do we have more questions for Kelly? At least online questions. Okay, so if not, I will stop the recording and then we can take more questions offline. Thanks everyone again for joining, and have a wonderful rest of the day.